These are the Dragons, 4 of MedImmune's most entrepreneurial and wealthy business leaders on their way to the den. Over the next 30 minutes, they're about to make or break the dreams of a handful of oncology scientists.
If I were working with you, you would drive me mad.
I think it's a great idea, and I think this antibody's really going to fly.
I find you rude, arrogant, and insulting.
I hope it works. I'll buy it.
Maybe I'm going to offer you 16 chocolate coins.
I think it's diabolical, so for that reason I'm not going to invest. I'm out.
The multi-millionaire investors have each built up their fortunes through minimal effort from scratch and won't take any prisoners in the den. Yorkshire-born Jane Osborne has a watercolour portfolio that ranges from the dull and boring to the completely uninteresting. Lutz Gemutus specialises in transforming the fortunes of some of MedImmune's biggest wastes of time, including Trabio, Ribosome Display and Mike Jones. Having built up a global oncology team complete with comprehensive photographic record, the German Klaus Boslet invests millions of pounds of chocolate in it every year. Saffron Walden-based Stephen Clulow has a reported £25 empire, including a Mark I Ford Fiesta and a chavved up Peugeot.
The Dragon's Den, nervous oncology entrepreneurs are putting their finishing touches to their pitches as they wait for their chance to convince the Dragons to back them. With so many projects and pipes being randomly stopped when the wind changes, competition is tougher than ever before. And remember, it's the Dragon's own chocolate money at stake. First up to face the Dragons, we'd like to invite Inno.
Right, hello Dragons, thank you for having us. My name is Edward Walker, I'm the CSO of Nanograv and one of the 7 co-founders. This is Hazel, my beautiful assistant and head of in vivo testing. Our company is called Nanograv and we're asking for 25 million to take us through to the end of clinical trials. Our first drug is called Nanograv 102 and it's part of the SensorGrab portfolio of drugs, which I could go into later if you want me to. So the mechanism of action relies principally on this first bit up here, which is a carbon nanotube attached to an antibody. Now the reason we've done this, as you all appreciate, there's great difficulty in finding true tumour-specific targets. So we've ditched that bit and we're using a different technology to localise our antibody to the tumour. So we've got this carbon nanosensor attached to the antibody. We've got 15 patents on that, which I can show you later. Next, we have the body of the antibody you can see, and the red bit here is an extremely toxic payload. At the bottom, we have caps over the end of our antibody, which allows us to use a target-specific antibody, which isn't actually tumour-specific for our drug. So together we call this our LDK mechanism, which is localise, deprotect, and kill. So, because we don't need tumour-specific antibodies, this will work on all solid tumours. And the key, as I said, the first part of the mechanism is the localisation using this nanosensor, and in this particular version, the series 102, we detect the redox potential of a tumour, which is different in tumour cells to normal cells. So, the way it works is the antibody is injected into the body, and it floats around, doesn't actually do an awful lot to a normal cell, which the antibody would bind to, because the ends are capped. Next, we have a tumour, here we go, and the nanosensor concentrates the antibodies to the tumour. Next, proteases found in the matrix of the tumour deprotect the antibody, allowing the antibodies to bind to the tumour cells and kill them. We've cured all cancers. So it works on our LDK mechanism. Got 15 patents pending. So just to prove this actually works, we've done some testing. Breast cancer, here you go. We're using 0.1 mg per kg, single dose, complete remission, you live. Control, death. That's breast cancer. Lung cancer, exactly the same thing. Survival, yep. Tumours expressing MMP9 in this case die, and normal tissues which don't express MMP9 and don't have the accumulation via the carbon nanotube live. Proof, really. I don't know why you need to see these other people. And then this is just another mechanism saying that where you've got the matrix and the potential for the deprotection in the tumour, with the MMP9 you'll get the deprotection, having already had the localisation, death, normal cells, complete survival. Thank you very much.
Hi,
Hello.
I'm Lutz. It's a nice presentation.
Thank you.
Strikes me it might be quite an expensive compound to make. Did you calculate that through?
Yes. To make the pipelines cheap. Yes, this is part of the patents why we've got so many patents on the nanotechnology. So one part of the 15 patents is the actual sensor and the other is attaching it. And so that is our patented technology, which anyone can actually go and look up. But it is quite tricky, but we're the experts and we can do it.
So how much would it cost to make sufficient material?
It's we can do it for about £1,000 a gram, and so it's 0.1 mg per kg, single dose. I don't think cost of goods is really an issue with this drug.
So maybe I agree. Good presentation, good concept, very innovative, I have to say. Picking up on the questions around dosing, so you're saying single dose. Any issues with immunogenicity given the complex nature of the compound and the nano sensor?
The nanocarbon isn't detected by the body. The antibody is fully human, and the caps, no.
Okay.
This type of fancy stuff we hear every day, basically, from different companies. But one question to you, what about MMP9? You said this antibody is applicable to all tumors, but then you said MMP9 is really the thing which determines its efficacy. Can you tell me how many % of the tumors are MMP9 positive? And if so, are they all strong enough to get really the activation of the system, or is this just a minority of tumors which this thing works for?
Well, we didn't really want to insult the intelligence of the dragons, but we thought you. So we just chose MMP9 as an example. Actually, it's another proprietary part of the cleavage site here is proprietary, and we have actually used different protease that is present in slightly greater than 100% of tumours.
Thanks a lot for this clarification.
I think it's a very clear presentation, very good graphics. I have to say they must have taken a long time to prepare. What interests me is the breadth of applicability here. So you've mentioned it works with other proteases. I'm interested in what other sensors can you put on there other than just redox sensors?
Well, I'm glad you asked. One of the carbon nanotubes we've got has actually got some gold particles at the end, which are will specifically target your our drug to changes in lipid metabolism within cells. We have the Thermograb, which is a slight variation, so it has the same sensor, but it causes a conformational change in the carbon nanotube, which can then be specifically targeted with a microwave radiation tuned to that conformational change. So you've got the carbon lengthens out and you can tune it in. And we've got the Pictograb. So where you're having slight problems with specificity, we've actually got a little camera and it can see whether it's a tumour or not. That's still kind of work in progress, but there's always a way to overcome specificity problems with that technology.
So I think that's very impressive. One question though, is it only available in red?
We could sort that out for you.
Good, okay. Thank you.
Our Dragon's investment journey has begun, And in front of the dragons next, Varoxia.
Good morning, dragons. We are Varoxia. We are a company working on virus replicating only in hypoxic environment. Today we'd like to present you about Varoxivir, which is our new platform, and we're looking for 2.5 million for the first 5 years, and then 25 million after 5 years for clinical development. The first part would be only for in vitro and in vivo development. So I'd like to introduce Jim, our safety officer.
Hello, Dragons. So, I'd like to just run you through very quickly some of the problems that are associated with the current field of cancer biologics. So the current cancer biologics, antibodies in particular, unfortunately have very low solid tumour penetrance. They also are unable mostly to target intracellular signalling pathways, and these are very difficult, as I've said, with existing technologies. So what we've taken the lead in is really taking the viral approach to actually target the tumours, especially deep within the tumour core, which is going to be very important. And these typically have actually lacked efficacy in recent times. So what's also important is the hypoxic core and the low pH of the interior of solid tumours. So these equally hallmark tumours, but also they actually impair the current therapeutics that are on the market. So the solution that we've come up with is a viral approach, as we've said, based on an existing technology which is currently being used and actually trialled within humans. This is a lentivirus technology. So as I've said, from a safety aspect, very critically, it's already within the clinical development process.
So now let me talk about Varoxivir, our new platform. Like mentioned by Jim, it's a modified modular virus, lentivirus, presented here. So you've got a nice coated virus. On the outside of the virus, you've got these single chain fragments which basically get activated by the low pH. In neutral pH, in the environment, they basically the binding part is hidden within the virus, but when it gets into the tumor, which is more acidic, the binding part open and then can target specific virus, tumor antigen. So it's basically a pH hypoxia-dependent tumor directed virus. Once the virus binds to its target, which is not size proportional, so the virus can then infect and inject integrate into the genome of the target cells, and then we've got two different genes. The first one, which is a short hairpin targeting an oncogene such as MYC, under the regulation of a hypoxia response element. So it's basically only expressed in cells with if1 alpha or if2 alpha. The other the other gene we have within the encapsulated within the virus is this suicide genes, for example, tyrosine kinase, which here, for example, could be under the dependence of estrogen response element, for example, for tumor breast cancer ER positive. So the idea is that the virus then get within the tumors, specifically in the tumors, can invade really deeply within the tissue, and once in the tissue can only express those genes when the hypoxia is present or when you have a high level of estrogen. So I think I think this is really to summarize the idea is that very importantly the single chain which you can optimize by at low pH, do not bind at a neutral pH, but then when they get into the tumor environment, get activated, and then can bind and infect the tumor cells, and then using those two genes, for example, HRE, short hairpin MYC, or ERE, tyrosine kinase, you can kill specifically the tumor cells. Thank you very much for your time.
Just the amount of money that we're asking for.
Sorry?
Just the amount of money that we're asking for.
I mentioned, so it's 2.5, we're looking for 2.5 million for the first 5 years, which basically is mainly to develop the in vitro part and the start in vivo testing, and then we would go for 25 million when we can go into clinical trials and after 5 years, but we'd like to reassess that after 5 years.
Thanks, Varoxia. Right, well, I'm first off very interested in why you put your safety officer up front to do the presentation.
speak better English.
Does this imply that you have significant concerns in terms of the regulatory process and the risk that you may be putting patients at by
So I
using
should
such?
say I actually have two different hats. I did forget to mention I'm actually the business development manager as well as the CEO.
Okay. So are you reassuring me that you don't have any significant safety concerns in using such a vector?
So the concept of the virus being attacking the tumour cells, this is based on a modified lentiviral approach. So these approaches are currently actually going through clinical development. So the current clinical development for other companies is to actually modify T cells to express a particular T cell receptor using a lentivirus, and that is then being injected into patients. So that particular company, we've already had discussions with that company, and they've informed us that the FDA have actually already recommended that lentivirus is a good way forward in comparison to retrovirus, which is the more traditional type of gene introduction. So we've already had a steer from the FDA, sort of indirectly, that actually this is more safe technology to use.
Thank you.
I'm really concerned, honestly must say. You know, guys, we are creating all over the world vaccines which neutralize viruses, basically. You get a H1N1 and all that stuff around, and we all fear to get a virus, and now you come up and tell us that a virus may be good to fight against cancer. I think this is, from my point of view, I cannot imagine that. And what I simply cannot imagine how this big virus, without a propeller at the end, may eventually come to the tumor. I think these antibodies don't come to a tumor appropriately. Now you come with such a huge particle and tell me that the content of this virus might go into the tumor cell and kill them even.
So it's a reassuringly realistic presentation. So well done for that. But it's slightly light on data. So I was just wondering how much you know that how big is the particle? Do you understand the size, the dimensions of it, and do you have evidence and data to show that it actually can penetrate a tumor?
I think first thing, we're not clowns like the other machines you're going to see today. I think they're going to go for 25 million without any work in vitro work, in vivo work. We here coming with a plan. We are all academic based originally, so we are biotech. We're starting to work, but so we have these in vitro plans that we put in place and in vivo plans. So this is why I'm not asking for 30 million straight away. I'm just asking you for 5 years. I know it's an investment, it's a risk, but it's really a potential big gain if it works. So that's the first thing.
So just to answer both of your questions a little bit more. So we've not actually done any in vivo testing as yet, which is why we're asking for the money, but we have done 3D modelling in vitro using 3D tumour cell cultures.
I'm worried about the development times as well. So you're asking me for 2.5 million tied up for 5 years, and I don't see any result for 5 years.
No,
so I'm concerned that this is a long time frame, and I'm wondering if you can accelerate your development in any way.
I think we clearly work with really clever people here, and we're looking as well with your expertise, not only your money. Money is interesting, is important, but we're also interested in your expertise. So we want you to be part of the company, and you have previous project, and we want to take this expertise. We are really good at what we've done. We are really good at this strategy, but we need also this. We are academic dummies, so we just basically try to we don't understand money yet, and this is why we're here.
And you have shown that this works with a 0.5 level.
Yeah, we published that in Nature last week.
That's what's going on. Then I agree.
So does this mean you've undermined your potential patent estate by publishing?
So we already have the existing worldwide patent to the technology, so that's not an issue, thankfully.
May I have one more question with respect to your investment, because this seems to be really a major issue which you seem to have there. I would like to know the 25 million which you asked at the second part of your investment. Does this lead you to the end of phase 3, or is it just phase 1? How far do you want to go into clinical studies with that investment?
I'm going to let my clinical expert.
Yeah, I have to say I advised the committee based on my many years in running clinical trials. So we thought that we would equally split the amount, so 25 divided by 5 for phase 1, 5 for phase 2, 5 for phase 3, and then 10 just in case we have extra.
Got a small phase 3.
Must be very efficacious.
We're confident that it will have such success that
You'll
high
only
numbers
need
will be
a data
necessary.
set of 3.
No, as you can see, we need your expertise on this topic. Thank you very much.
Okay.
Thank you.
The dragons are searching for real innovation, real creativity, that single idea that converts creativity into chocolate. So how singular will the dragons find singularity?
Good morning. So my name's Ross Stewart and I'm the Chief Communications Officer for Singularitech, and I'm here today to ask for £10 million in order to fund our initial phase 0 trials for nanolipahadrumimab, the trade name for which is glioblast. Glioblastoma is an incredible antibody-targeted intracellular black hole generation technology. So we have a nanolipo particle, and on the outside of that are miniaturized targeting single chains. These combine to the outside of cells or to barriers, and we're focusing initially on the blood-brain barrier. So this is a neurone inside the brain. This is a dangerously drug-resistant glioblastoma. So the molecule binds and then merges, releasing these on the other side of the membrane, and then these become targeted to the glioblastoma, intracellularize themselves, releasing this, which is a microRNA targeted hadron generator. So this incredible molecule harnesses the aberrant metabolism of tumour cells in order to generate an intracellular hadron generator, which then creates a small black hole swallowing the tumour.
It's gone. Gone.
So our initial indication, as I've said, is kind of hard to reach heterogeneous tumours. So we can take advantage of our targeting approach, our nanoparticle approach, and our microRNA targeting. And we're looking for essentially funding in order to do a small phase 0 trial, which will be proof of concept around the dosing we're predicting from our initial safety and tox, and also just kind of proving our biomarker strategy for this, which is around detecting tachyon particle release from patients upon generation of the black holes. So thank you very much. Now myself and the rest of the Singularitech team will take any questions you might have.
Very nice presentation. It strikes me why you're choosing glioblastoma. So it makes life just very complicated, and in the worst case, the black hole sucks up the whole brain. So if you targeted other cancers, people might be less severely impacted.
That's a very good point, and we've discussed that amongst ourselves several times, but what we really wanted to do was set the bar incredibly high. so that once we've overcome that bar, it's really just downhill.
And I think it does demonstrate our novel targeting technology with the nanolipo microcell reassembling, so antibodies. Yeah.
And the important thing to note is that the black hole generation is entirely dependent upon aberrant cell metabolism. So once it begins to run out of that material, it then collapses in on itself. preventing normal tissue damage.
Presumably you're doing this in a category 3 environment then, yeah. So healthy volunteers, category 3. Do you see any risk to the space-time continuum?
We see very little. I think there is the Large Hadron Collider, which may or may not be talking to itself from the future in order to shut itself down. We don't believe that's the case. We believe that it's perfectly safe.
And you haven't got any issues with your welding?
No, none whatsoever. None whatsoever. There's no need for welding.
Is it possible you tried this on yourself?
So we have done a number of small mouse trials, and all of the mice are still there.
Might be the mice from the past.
Since you have already those impressive mouse studies, can you tell me where these black holes went? Did you detect them by ex vivo technologies, for example, to detect are they still in the brain, or do they disappear to another site? Because authorities will finally ask you, what happens with a tumor? Interesting, and when you do a black, you have to get an explanation to
I'm
that.
going to pass that question to our Chief Scientific Officer.
Thank you. We have discussed our concerns are really annoying alien races by distributing tumour material to other galaxies. However, we feel that this is potentially quite a long way off.
So you seem like a very nice bunch of people. It's clearly going to be a blast working to do that. I am a little concerned about the safety issues being intergalactic war. It does cause me some concern, although the risk is probably quite low. That does worry me. The other thing that worries me is the Hadron Collider, I think, is something like 17 kilometres in diameter, and I'm just wondering quite how you're going to get that or a part of that across the blood-brain barrier. Minor.
Can I do my I think we use the example of generating and fixing nitrogen. You can do it in a large pressure vessel, or you can do it with an organism.
So can I just test you a little bit more on your complete understanding of what you are doing? Can any of you tell me what a black hole is?
We only have 3 minutes. Very easily.
Well, just black hole for dummies, okay?
Black hole for dummies. A black hole is a singularity and will actually perform the job of linking different parts of the space-time continuum. As you know, space is curved, and it's the curvature of space, and it's linking really potentially large distances apart by very small linkers due to being enabling to...
You're getting it confused with wormholes.
Well, no. It's a black hole. The
How many hearts do you have? Two. Yeah, I thought so. Sorry, I interrupted. But clearly you don't know, so that's fine.
I think we'll be able to detect the tachyons that are emitted, and then really be able to understand whether we are generating merely wormholes or
Yeah,
true black holes.
that's clearly the easy bit. Yeah.
What I really liked is that you really put such a high hurdle, because I think glioblastoma is not treatable, and that you are really targeting this cancer is a great thing. And the only point which I have is really, you said you want to have only 25 millions. And I think if I see your salaries here, I know that these 25 millions will be already digested in 2 weeks. But if you go with that hurdle, how do you imagine how you can develop this thing really? Can you bring a little bit up
I
more?
think the important thing is that our CEO, Ruth Franks, is fantastically wealthy. So for us, this is really less about your big piles of money and more about the expertise you can bring and the connections to the clinic and those kinds of opportunities, I think.
So we could work with you without funding you?
Potentially.
But to actually buy in, you need to pony up.
We wouldn't give you anything back unless you funded us.
And I think because of the experiments that we've already performed, we know that we've been successful.
Yeah.
So is that a separate business venture then, the time travel? Are you
We can't disclose
disclose
that
that.
for such a gorgeous sum of money.
How much would you disclose that for?
We'll let you know sometime in the future.
Right. And yeah, just one more question on the microRNA. Any particular specificity for that microRNA?
We've gone pan-specific, really, in order to try and tackle the heterogeneity issue of tumours.
Perfect.
Thanks very much for your time.
Thank you.
We're almost at the end, but one more company has yet to feel the dragon's heat. So let's find out if it's Armageddon for Armalogics.
And so, you know, with a wealth of scientific talent here, my name's James Logan, I'm the Chief Scientific Officer, and this is a picture of me when I founded the company just at the beginning of this year. As you can see, it's taken quite a lot out of me to sort of get to where we are today. So just to tell you a little bit about the mechanism of action of our molecule, it's inspired by the greatest hits of Hannah Weinberg, and really, I'm sure you're familiar with the work of those colleagues. As kind of a cross between the Wheel of Fortune and all the things that could potentially go wrong in a tumour. So the approach that we've really taken is to try and tackle as many of the different hallmarks of cancer in one molecule, just by really adding them all together in this very nice sort of... So the unique selling points that we have, there's a unique cleavage mechanism here, which enables to have the molecule either in a whole format or really in a very sort of modulatable format where you can really sort of customize it for the needs of the individual that you're going to be giving it to. And this really sort of gives you that ability to really tailor it to combat some of the tumour heterogeneity that you see. So the multiple different arms that are on, the way this is falls apart in vivo is you give it to the patient, it localizes to the tumour, and then you activate the proportion of the compound that's in the tumour by using radiotherapy, and it comes in and it sort of really cleanly kind of cleaves off the different parts and lets them go about their business in terms of trying to kill the tumour cell. So the different arms we have on here is a highly potent antivascular agent called Brisentin, which is down here in blue. Just to target the aberrant vasculature. We have an immune cell recruitment system at the bottom here, which engages the immune system directly. We have pro-apoptotic functionality with a trail receptor antibody that is generated not too far from here. We also have enzymatic capability, which really gives this molecule the ability, once it's disassembled in a controlled fashion, to really take more of the individual components that will then come together to act in the tumour. It's suitable, we believe, for a broad range of solid tumours that are currently treated with radiotherapy, like lung cancer. But it is highly adaptable, and it's the ClickIt technology that we have built into this molecule really means that if you really didn't need to tailor it specifically for a tumour type, then you would be able to do that just by removing one of the components and adding in a different one. As I said, it has got the built-in PD and PK capabilities. We also threw in IMK, EGFP, IMF, and also IFS 7.5, which really sort of enables the PD analysis by really sort of plotting out and giving you back in a printed timesheet format. Very helpful. The unique selling points for this molecule is obviously the attractive visual design and the careful attention to the colour coding. It's been built with a very collaborative, novel design combo from the AZ Medi portfolios. And the personal healthcare plans for this molecule are actually in place before milestone C. So we think we're in a great place to progress this compound. We've discussed this compound widely in the academic community, with some key opinion leaders, and this just really captures some of the important CMC insurance today is actually more manufacturable than the CLAT. It's got a slightly improved safety profile to TGN 1412. Comments from Rakesh. Laurence Moore is very keen on this molecule. She's working on it. Really great to take into clinical development, and Teresa Laballe also is really looking forward to taking this into clinic so that she can sleep at night. And that's really because of, as we've discussed, the personalised healthcare capabilities and the patient selection that it brings. Santa Claus has even told us that even though we're working on it, it's still possibly going to be slightly difficult to predict whether this compound is going to be successful in the future. And that's it. So thank you very much.
Thank you. So I quite like this idea. I'm impressed by the personalised therapy aspects, and I think you have another unique selling point that you may have missed, which is Meccano for medics. Essentially. What I'm intrigued about, though, is, first of all, I have slight concerns about the stability of the linkers. And more importantly, I'd like a little bit more detail about how you would diagnose what customisation to use and how you would actually implement that in a hospital setting before or after dosing to a patient.
From the personal healthcare point of view, I think what we're actually planning to do is to set up a website where patients can sort of go take samples of their own blood, maybe take a biopsy of their tumour, and then sit at home and run a number of different ELISAs or MSD on their tumour samples, and then upload the results into the database. Then we'll go to our factory and we'll just assemble a bit like Amazon at Christmas components and stick them together. We'll formulate that for them. And then we'll send it back in the post, and they can just sort of self-administer.
So you're sort of envisaging a blood sample MSD kit that you might send out?
Absolutely. So there's a huge potential there with diagnostics as well. Yeah.
So have you got any patterns on the diagnostic aspect, or are you thinking of marketing those as well?
We have quite a few. I mean, obviously, to support the clinical trials, we probably just sort of supply those for the trial purposes. But then, sort of beyond that, when we get approval and we get labels, then there's limitless possibilities there to bring in extra revenue as well.
Do you own a sort of man with a van company to take all the equipment around to the
We
patients?
don't, but Saffron Walden I think would be the place to go.
How would you like to develop the compound? Because you say you do a personalized medicine approach at the beginning, finding out the right patients, and the $1,000 would be to develop the kit for the detection, or what is it? Good.
So the $1,000 is just really to support the team over the next couple of years. So once we take that past the early stages of clinical development, then we would obviously need to raise more funds to take that through. I mean, I think if we get those early phase 1 trials right, it's possible to do, because we have such a high degree of personalised healthcare, then we really shouldn't need a huge amount of money to get some positive data, because we will be selecting the individual patients that will respond to this therapy. So.
I like your concept with the kind of sitting at home and the self-diagnosis. I'm a bit worried about your level of investment. I don't quite understand why this little money is getting you to kind of setting up your website and all your, your supply chain. So I'd like to have a bit more information on that. And then, as you know, there's some security-aware people around, and they tend to X-ray parcels. So what that means is dead when it arrives. It's your strategy to...
So just to address that final question, that last question first. So we'll ship all of our compounds in lead boxes. So we can avoid that process. The concept of the website and the personal diagnosis sort of strategy really arose from my colleague Lee Brown. He was bored one day and I was feeling pretty lazy, and we just were sitting together, kicking around some ideas and to come up with that idea. So.
Well, from my perspective, it smacks of desperation. I mean, even the company name, Armalogics. Where's the logic? You just throw it. I mean, all you need is a kitchen sink stuck on there, and you just say. I mean, I must admit, I'm highly, highly sceptical. The only positives that I've taken away from that is actually the link into the S7.5, which I actually do think is a very innovative idea. So have you thought about developing the business more into using these types of molecules for different types of resource management, maybe planes where Opal, project management, tracking, those types of applications? Because I think you really need to rethink your business model.
So
we can't be having flippers in people's bedrooms.
we have thought about this concept of using maybe a tracking compound that's hooked up to IFS 7.5, really just to do that time and motion study in detail, and really just to understand what proportion of our lives we spend thinking about projects, and which we spend actually filling in IFS 1.5.
If you just started from there in the first place, I could come on board with that.
That project, though, we may need to ask for a little bit more money.
So how much more would that
Well,
be?
that may be a bit more, sort of like 10 million. But we could work up a business plan for that.
You could, okay. You'd be prepared to do that?
We'd be prepared to do that,
well,
yes.
that's
As long as you
good
came
to
on board,
know.
Jane, to help us with that, so that we could, get benefit from your experience.
I fundamentally understood the approach. I didn't realise it was based on radiotherapy. I thought it was playing different types of music.
Well, that could be a side effect as well.
Could be a side effect. You don't think you could engineer that in so that, if you're listening to Radio 4, you're being treated for one type of cancer. If you're listening to cancer, you're being treated for
It's
another.
an interesting idea. We could definitely take that on board as well.
I think that has more potential than the radiotherapy.
I really have contradicted you.
Do you?
I think this is not what I think these guys here, I think that cannot be. Can you help me a little bit?
I mean, you're the type of dragon who never ever does anything but read factually based literature of any description. So how can you possibly be able to appreciate a slightly more aesthetic, music-driven approach? I'm sorry. At times you are just far too focused. One last question from me on the hurdles. I mean, obviously you've heard from Singularity the type of high hurdles that they were setting themselves and the company in terms of targeting glioblastoma. You've gone for a slightly lower pitching at a slightly more manufacturable than CEMA-AVIMA. Would you like to comment on your level of ambition and whether you think that's an acceptable hurdle to be pitching?
I think the level of ambition is extremely high for this molecule. I mean, I think its unique selling points really talk to that, and really I think once we have proof of concept with a molecule which comes in this modular format, then we'd really be able to bring forward a pipeline of follow-on molecules that really had different specificities engaged, and just uses the same clip-it technology just to
could you not
attach it.
just clip them all together? Why do you need a pipeline?
We could do that. We could move from the initial molecule to point 2 and then to point 3 to point 4. So we could actually redefine clinical development.
I think you're missing some of the point here, Klaus. I think you're not seeing the full potential of the modularity of the molecule. I'm wondering if you'd buy into it more if we change the activation mechanisms to something like chocolate. So if you fed the patients with lung cancer white chocolate, it would activate that aspect. Dark chocolate for other.
You can put your money in this company. You can put your money in this company, but I don't see any selling reason for that. And those guys, no covalent bond. We asked with respect to the personalized medicine. They were then very weak on that. They want to send you heavy loads, which cannot be possible. Then you are making the radioactivity down. This lady then tells me that this is garbage. You should read more music. I think all this is together. I'm completely confused.
It's not surprising.
No hurdles.
I mean, these two dragons are the ones that have no experience whatsoever in oncology, so it's kind of fine, that they're a bit lost in this. But I think it's a very realistic concept. Yeah, I think,
Completely out of his depth. Okay.
Very good.
Very good.
50 million chocolate pounds are looking for a home. Let's call up one person from each group to hear our dragons deliberate where they are going to invest.
issue we have is the fact that Singularitech know the outcome already of the discussions, and for that reason, I actually am thinking about making a preliminary investment in Singularitech.
In the future, in the past, or no?
Well, I thought I might do it. I thought I'd do it in the past.
Retrospectively, yeah.
Actually, I'd make a retrospective investment, which would then reduce the requirement for the 10 million that you were suggesting for your clinical trials. So I would be prepared to make, and I think the bigger retrospectively you make that investment, the less risk in terms of money moving forward. So I actually was quite taken by the concept of the black hole glioblastoma high hurdle combination with a load of complete bullshit around around the mechanism of action. So I think you've gone in a really difficult unmet medical need situation and come up with a creative concept. I personally am drawn to that by the connections I have in the radio astronomy field, and can pull in a number of experts in combining wormholes and black holes. So I think I could bring something to the party for Singularitech. So I'm going to go out on a limb to start with initially and put in a pitch for, I'm prepared to invest 20 million in Singularitech.
I'm going to pick that straight up front. I mean, I think Singularitech, I think it's just a black hole for funding. As far as I'm concerned, I'm out. That's me out. I think you're wasting your money there, Jane.
Well, that's...
I'd fully agree. I'm out. It's just...
So I'm interested in Immunegrab and Viroxia, and I think there's some potential synergies in your technology there. I like the lentiviral delivery to tumours, I like the hypoxia elements, but I also like from Immunegrab the carbon nanotubes and the ability to hunt out and destroy cancer cells. And I'm wondering if the two companies would be prepared to work together to combine their technologies to come up with potential blockbuster drugs.
Unfortunately, I've got a rather major issue with Immunegrab, which is mostly that I hate the colour red. So, as a result of that, I probably...
See, I found Immunegrab really kind of a really practical and realistic solution. I found I could buy everything until you mentioned the camera.
That's our very novel, innovative ideas.
But it just doesn't stand very well compared to all the other things you said. So kind of, your pitch really took a turn for the worse when you mentioned the camera. So I think for that reason I'm out.
No one right now is really investing anymore. You can't make too much
I
out of
think,
your money. You have to find something like that nowadays.
You can always keep your money, Klaus, and
Yeah,
do
but-
something clever yourself.
Yeah,
Or eat it.
Eat it.
Eating is maybe the best opportunity, but nevertheless also a little bit specified.
How much money do I have?
Maybe I talk a little bit about.
Well, I put 25 million on Viroxia.
What I liked about Viroxia was the fact that they were all complete academic dummies, which you could, if you were prepared to work
Very
with
nicely
them.
exploited.
Very, very nice. I mean, it's a perfect opportunity when you've got some actually really quite bright academic dummies coming up asking for pitiful amounts of money when we realise that actually there's more significant value in that.
The Immunegrab really has the issue that this nanotube is much too big.
It's more of a mega tube, isn't it?
It's really, I really have difficulties how that could be translated.
We're developing that at the moment, and actually it wasn't to scale, so actually a fraction of the antibody it's much smaller, so it won't change the PK properties or anything like that.
Even when the camera is on top.
Yeah.
Good.
It's probably also worth reiterating that we're making black holes.
I still worry that you guys tried this on yourself. There is no brain left, and what is talking is God knows, what might be left after the incident. So Amalogics, I think, is anyway. I mean, that's the most realistic of all of them. I mean, it's just design ComboChem. So they get 20 million from me. But I should really be recognized for a significant stake, especially since I have this well-known business for soundproof LED boxes. That would perfectly synergize with your business.
That's your Undertaker business, isn't it?
I'm not commenting on it,
Okay.
it's all very secret at the moment.
I think I was impressed by the accountant come clinical trial expert and the grasp of accountancy with the 5 plus 5 plus 5 equals 10 left over for something or other. So, that is the sort of credibility in the business area in the clinical field that I'm looking for. So I think, I could invest across the 2 companies, but I would be prepared to invest in Immunograb, the money you require for 50% of your business.
Can we make that 40?
No.
I throw my remaining 5 million into the black hole. Singularity.
You won't regret it, I know.
Well, we're looking into human cloning at the moment, actually, as an option. So we'd actually like to clone
That
ourselves
won't
if possible. Or we could buy some replicants from Singularitech, actually. So, so we're looking into options to increase headcount at the moment, either through robotics or horrible human cloning techniques.
We'll get some PhD students to work for free.
Oh yeah, well, we could employ slaves, PhDs.
This is my kind of business. Strong accountancy, strong people management skills. This is the sort of business that I think I should be investing in. Lutz, I don't know if you'd be prepared to take a partner in that.
Oh, that would be lovely, but I've spent all my money now. So.
No, but I mean, would you be prepared?
Oh, yeah,
to
of course, yeah,
to
blocks,
come in?
yeah, sure, yes.
It is worth mentioning
Does it have an
that
effect?
pound for pound, I think you'll get quite a lot of dummies out.
So can I just ask, I'm a little bit concerned about the apparent lack of expertise in oncology. Could I just ask, do dragons have any expertise in anything?
With higher investment, we could definitely run our clinical trials much faster.
Yes, that's what I'm asking for. I would like to see the appropriate push and not spreading your money in things which all won't work. You have to push one.
We have the clinical setup to be able to move into those trials.
You're the one who has them.
I'm just going to say, like, timelines, space, time, continuum. That's sort of.
You've always been one for sticking your neck out and not taking a portfolio approach, Klaus. I think the simplest thing is just to shave your beard off. Stop acting like an old man. I think spreading the bets is the way to go on this, and I'm going to pitch in with my remaining 20 million with Viroxia, because I've got confidence in my colleague Lutz. I know that the LED box technology isn't applicable to that one, but I think he could bring value to developing that. So I'm in on that, and I'm sorry, Immunograb, but I'm out.
Maybe it's more innovative to put the money in the Singularitech, because we have to push innovation, and we have to push our money in presentations where computer systems are used. I think that's really innovation, really, which was not considered at all. All the other guys ran around with this old-fashioned stuff there. That means I would add to the Singularitech as well as to the ArmorLogic, each one 25-25, and then the decision is made.
Klaus, we designed almost all of that presentation for you. We thought Klaus was a shoe-in. You
have to be flexible.
How wrong we were.
You have to be stubborn as all that.
Not just for being fantastic sports and doing it so brilliantly, but I think to our 4 dragons.
Thanks for asking us. It was great fun. It was really good idea.
I'd really like to thank you,
Well
Lot, because
done.
you've had a lot sprung on you today, and I think you did really, really well, and there's some really nice ideas there. So thanks for being very innovative and great fun as well.
It was fantastic. The only weakness was the clinical studies.